So we are Iju.
And Sehi.
Today we'll talk about the technique that has developed for the past 10 years. The technique is called embryonic stem cells, also known as ESC. It is isolated directly from the inner cell mass at this blastocyst stage during implantation. Blastocyst is an unimplanted human embryo at the 6th to 8th day of development. These cells are pluripotent, which have different genetic makeup. Donor cells are harvested through biopsy. Mature somatic cells are reprogrammed to pluripotent stem cells, which is given the appropriate chemical signals. 4 transcription factor genes encoding OCT4, SOX2, KL4, and CMYC are used for the transmission. Chemical signals in form of transcription factor or pluripotency genes, also known as iPSCs and ES cells, are transplanted into people with different genetic makeups. ES cells have some clear advantages over adult stem cells according to the concerning research and clinical usefulness. For example, ES cells are pluripotent, meaning that they have the potential to give rise to all types of cells in the body. Yet adult stem cells are multipotent, meaning that they only have the potential to give rise to a limited number of cell types. So far, no adult stem cells have proven to be pluripotent. This means that ES cells could potentially provide a renewable source of replacement cells for any tissue in the human body. ES cells are also relatively abundant in the developing organism, especially compared to adult stem cells, which are scarce in the adult body. As a result, ES cells are much easier to identify, isolate, and purify compared to adult stem cells, which are very difficult to identify, isolate, and purify in the lab. This makes research with ES cells all around easier than research with adult stem cells. However, there is negative implication related to economic. iPSC, which is induced pluripotent stem cells, costs per line $4,500 to $9,800 per cell line. Moreover, embryonic stem cells cost per line $825 per cell line, which are very and highly expensive for normal citizens to afford. However, we think that this technique is beneficial to mankind because there are recent researches that are active on embryonic stem cells, and the economic factor can be reduced and solved in recent times. Thank you.